Hello. Okay, so let's see, a couple of announcements. I had to redo the review session. Unfortunately, I cannot do the review session on Saturday night. So I decided to go with later Friday night to accommodate those who are going to Portland and then coming back. So Friday night, the review session will be at 7 o'clock in ALS 4001. Okay? 7 o'clock, Friday, ALS 4001. I will videotape that if you can't be there. And if you can't be there and you have questions, please come see me. I'll be more than happy to answer questions for you. After today, my schedule lightens up a little bit. So, and if you need to see me, come see me. I gave my phone number out last time, and so if you need to give me a call, I'll be happy to try to answer by phone as well. Some things are better in person, so I may not be able to always help you on the phone, but I'm always happy to try. Question? Well, the reason I don't want to do a review session on Sunday night is I don't know for sure it'll get posted by Monday, so that's why I go earlier rather than later. I will render the video Friday evening, and I would expect it would be ready sometime Saturday morning. But I can assure you I will work to get it done as quickly as I can. Yeah. Until the questions run out. It's usually about an hour. But yeah, I'll be there as long as you have questions. I'm happy to answer your questions. Okay. For the first exam, it tends to run a little bit longer because people want to see more problems worked and things like that. All right. So that usually tends to take a little bit longer. Okay. So Friday night, 7 p.m. There goes your weekend, right? There goes my weekend too, if it's any consolation. So, okay. We're in pretty good shape with where we are, where we need to be with things. And in fact, I need to leave a little bit early today myself because I've got to go to Portland tonight to give another talk. I think all I do in the evenings anymore is talk. So I'm going to try to finish today by about 4:45. And I won't be able to meet with you after class, unfortunately, today because I do have to get out and get up there to give the talk. So as I said, after today, things at least do settle down a bit. All right, so I've been talking about the ways that we control enzymes. And as I said in class, it's very important that we control enzymes because if we don't control enzymes, they might be making too much of something and wasting energy, for example. And if they're wasting energy, they could be killing the cell because if they're using energy to make things that the cell doesn't need, it could burn up all of the cell's energy and the cell would not have energy for other things that it needs. So getting enzymes under control is a very, very important consideration. All right? Now, I've talked about two different ways, excuse me, hiccups. Two different ways that cells can control enzymes. The first of those was allosterism. And allosterism, of course, is when a small molecule bound to a protein and affected the protein's activity. That was either positively or negatively. The second mechanism that I started talking about last time was by covalent modification. And so one of the covalent modifications that you saw was phosphorylation, dephosphorylation. They're both essentially backwards and forwards of the same thing. As I noted, phosphorylation can lead to the activation of some enzymes and the inactivation of other enzymes. So it depends on the enzymes in terms of whether getting a phosphate makes it active or inactive. Okay? The example I gave at the end of the class period, which I'll just show you here, was that of glycogen phosphorylase. And glycogen phosphorylase has a rather complicated regulatory scheme. I'll say more about it at the end of the year. But one of the things that I want you to focus on is the fact that the enzyme can be converted back and forth between a phosphorylated state and a dephosphorylated state. In the case of glycogen phosphorylase, when it gets phosphates, and you notice that it gets 2, when it gets phosphates, it tends to be more active. tends to be more active, whereas losing phosphates causes it to become less active. All right? Now, this enzyme is involved in taking glycogen in your liver and breaking it down and releasing glucose for your muscles. It goes into the bloodstream and the muscles pick it up. Okay? Well, how do we have glycogen in the first place? We have glycogen in the first place because something made it. Everything that we have in our body we had to have made. So that meant that there had to have been enzymes that made that glycogen. Okay? Now this is a prime example of where we want to be careful about turning enzymes on and turning enzymes off. Let's imagine in my little liver cell that I have a signal from the muscles that, hey, we need glucose. And the liver says, okay, we're going to put phosphates onto glycogen phosphorylase, and we're going to activate it so that it starts breaking down glycogen to release glucose. So little glucoses start being released. What if, at the same time, the enzyme that makes glycogen started putting those together and making glycogen? In other words, it didn't make it out to the cell, and make it outside the liver cell. That would not be good, because the muscles are still waiting for that glucose, and something is breaking them down, but then another enzyme is taking and making them up at the same time. It's a prime example where we don't want to have the breakdown enzymes active at the same time as the synthesis enzymes. Okay? We want these to have opposite effects. When we're making it, we certainly don't want to be breaking it down either, because that would be wasting energy, right? So we want to have a control where we make one where the synthesis enzymes are active and the breakdown enzymes are not active, and vice versa. And phosphorylation turns out to be a very, very easy way to do this. So I've got two sets of enzymes: glycogen making enzymes, glycogen breaking down enzymes. The glycogen breaking down enzymes are turned on by phosphorylation. The glycogen making enzymes are turned off by phosphorylation. One thing has opposite effects, which is exactly what you want those enzymes to have: opposite properties. One active, the other one not active. I'll repeat that. The glycogen breaking down enzymes are active when they get that phosphate. The making enzymes, the glycogen making enzymes, are inactive when they get a phosphate. Okay? Now conversely, when we take the phosphates off, the opposite happens. The glycogen making enzymes become active, and the glycogen breaking down enzymes become inactive. Does that make sense? Now we'll talk about this principle later, but this principle is a very important one when we get thinking about the chemical reactions that are going on inside of cells. The principle here is that of reciprocal regulation. Reciprocal regulation. I'll talk more about that later, but I think you can start to see the reciprocal part. The breaking down enzymes are active when the other ones are inactive by the same process, putting on a phosphate. Vice versa, when we take off the phosphates, the two are reversed. So reciprocal regulation turns out to be a very, very powerful way for cells to control and keep from having synthesis and breakdown going on at the same time. You don't want to have that happen. It's wasting energy. It could be killing the cell. It could be killing the organism if those are going on at the same time. So I hope that illustrates for you why we are very, very interested then in understanding how enzymes are controlled by cells. Questions on that? Yes, sir. Okay. Yep. That's right. And then the two get reversed when you take the phosphates off. Make sense? Okay. All right, good. Okay, well, there are other types of covalent modification that can happen to proteins, and I'm going to talk about that just a little bit. Okay. So one of the types of modifications is by the cleavage of peptide bonds, by breaking peptide bonds. Okay? So remember, we make peptide bonds by, that's what we put amino acids together to make peptide bonds, if we break peptide bonds, we are breaking the joining of those amino acids to each other. Okay? Well, enzymes that break peptide bonds are called proteases. Protease breaks a peptide bond. Okay? Well, that's cool. But let's think about this for a second. When do we want to break peptide bonds? Well, one of the times we want to break peptide bonds is I go eat a Big Mac, and I've got, you like Big Mac, okay? I got all that protein that came with that Big Mac, okay? And I want those amino acids for my cells to do something, so my digestive system says, hey, we want to break down this protein that he's just eaten so we can have the amino acids to make our own protein. Okay? So our digestive system is one example of a place where we have proteases that help us to, in this case, have nutrients that we need. Okay? It breaks down those proteins. It releases the amino acids into the bloodstream. Our cells pick them up. They can use them to make their own proteins and do their own things, which is very cool. Okay? Now, there's a problem with that. Okay? The proteins that are the enzymes, and by the way, somebody said last time I said proteins when I meant enzymes. They're the same thing. Okay? So the enzymes that we use in digestion are made in our pancreas. Most of them are. Most of them are made in our pancreas. Okay? So our pancreas secretes these guys into our digestive system, and then there they go and they do their thing. Now, the problem is that these proteases break down proteins. Don't they break down themselves? Don't they break down the cell proteins that they're being made in, et cetera, et cetera? You can imagine we've got a problem, right? So it turns out that our pancreas does not make proteins in the active form. It makes them in an inactive form. If it made them in an active form, you end up with a very severe problem called pancreatitis. Has anybody in here ever had pancreatitis? It's not uncommon. Pancreatitis happens when normally your pancreas releases the inactive proteins, the inactive enzymes. And by the way, an inactive enzyme is called a zymogen, Z-Y-M-O-G-E-N. The pancreas releases zymogens, and they travel down these little ducts and make it into the digestive system. When they get into the digestive system, the process that you see here happens. Digestion happens where there's breakage of peptide bonds in various places. And so we end up with, starting with one long chain, we end up with 3 individual chains joined by disulfide bonds. The yellow things are disulfide bonds. The disulfide bonds hold it together. As a consequence of these changes, the active site of this guy is opened up, and it can now start breaking peptide bonds. Whereas up here, the active site is closed, and it can't break peptide bonds. Okay? The pancreas released it in this form, and as it got closer and closer to the digestive system, the enzymes of the digestive system helped to start doing this breakage because they're active in the digestive system. What if this guy starts being active not as it enters the digestive system, but a little bit up in the duct? And then that in turn activates things a little bit higher in the duct, and a little bit higher, and a little bit higher, until finally we have back at the place where the enzymes are being made, or the zymogens are being made, we have active enzyme. Those enzymes are going to start attacking the pancreas, and that's what causes pancreatitis. When we have those active enzymes, those active proteases being present at the level of the pancreas. They attack the pancreas, they damage the pancreas. It can be a fatal disorder. It usually is not, but it can be. If you've ever had pancreatitis, or you've ever known anybody who had pancreatitis, they will tell you it's a very painful disorder. Okay? And it happens because this form is becoming present at the level of the pancreas and not further down in the digestive system where it needs to be. Yes, sir?
How would you treat it?
How do you treat it? Good question. So the way you treat it is there are protease inhibitors. A protease inhibitor is a molecule that binds to the protease and keeps it from becoming active. It is a fairly treatable disease, but it really, as you can imagine, upsets your digestive system because the more protease inhibitors you have there, the less you're able to digest. Okay? So